My task is made easier because the issue of residual risk has already been discussed quite a bit in this meeting. So we're here to celebrate how far we've come, but also to recognize how far we have to go. The battle is engaged, but it's not yet won. We've heard about this residual risk even in patients who are in clinical trials who've been treated with the latest and greatest of antithrombotic or anticoagulant therapy on top of contemporary anti-lipid therapy. You see, as has been mentioned, that the recurrent event rate in the first year following an acute coronary syndrome is on the order of 10%. Now, these are in clinical trials, and things don't get better than clinical trials. And if we go into the real world, into the Swedish registry, and this is a first world country, not like where I come from, with a medical care system that is very, very good and very aggressive. If you look at patients that are like the ones that I see, and maybe many of you see, that are elderly, that have impaired renal function, you see that the recurrent event rate in these higher risk patients that actually are what we see in practice, not like what we enroll in clinical trials, is up to a third. So 30-odd% of these people will have a recurrent event in the first year. So as I say, the battle is not yet won. So let us all remember, because we're all engaged in development of pharmaceuticals, that lifestyle is the enduring foundation of all cardiovascular prevention. But unfortunately, as you know, actually instituting and maintaining a healthy lifestyle in our real world is very difficult. So you saw earlier a wonderful picture of different categories of residual risk that was first authored by Patrick Lawler, who was a university student in my laboratory when he started out, and then was our cardiology fellow, and is now making a wonderful career in Canada, in Toronto. But this is my version of the slide, different baskets. And maybe this has never been published, so maybe we can include something like this in a publication that might come out of here, because I like these baskets of risk. And we've heard already about LDL, and we'll hear a lot more about LDL. But we also, and you've also seen a version of this earlier, that even in the people, as Bob Giuliano pointed out in this follow-on paper to the Fourier study, that there is a striking area under the curve of residual risk, at least over the time of inhibition of LDL with evolocumab, which we have to address. So my brief then is to go beyond the LDL story, as successful as it is, and talk about residual inflammatory risk. I'm not going to rehearse the inflammation biology in atherosclerosis from the beginning to the ultimate thrombotic complications. You've heard me speak about that many times before, and I don't have to remind this audience about that biology. Now, this is a slide that we will see several times during this meeting. It's a famous slide prepared by my clinical colleague and collaborator, Dr. Paul Ridker, who unfortunately is having to care for his parents right now and could not make this meeting despite really loving Marrakesh and Morocco. What he did here was to look at some contemporary studies and ask, well, recurrent events, how much of that is due to residual lipid risk defined by an LDL of greater than 70 milligrams per liter, and how much is due to inflammation as defined by the biomarker of inflammation, C-reactive protein measured with a high sensitivity assay, HSCRP, greater than 2 milligrams per liter, which is a cut point that is approximately the median of a population walking down the street. And as you see, the slice of the pie due to residual inflammatory risk, which is depicted in blue here, is greater than that due to residual cholesterol risk, as shown by the deep orange or brown slices of the pie. So this is a substantial part of what we need to deal with in our patients who are already well treated in terms of LDL reduction. So for the last 15 or maybe more years, I have spent a great deal of time, particularly on the clinical side, in trying to answer this question: can targeted anti-inflammatory therapy improve cardiovascular outcomes in people, and in people who are well treated with our contemporary lipid therapies? Now, there are different targets that we can use. There are cytokines that are the protein mediators of inflammation and immunity. There are bioactive lipids that we already heard about, reduce it and strength, and it would be wonderful to have some discussion during the roundtable of some of the issues with interpretation of those studies and how we can move forward in this lipid mediator. I might also add that some of the resolving mediators that have been pioneered by my colleague, Dr. Charles Sirhan at the Brigham, are a very important part of the other side of the anti-inflammatory spectrum, because the pro-resolving mediators may avoid some of the interference with host defenses that we see with some of the therapies that I'm going to be speaking about. And then there's reactive oxygen species. Well, we have thousands of papers about oxidized LDL, and everything works in animals, but the clinical tree has not borne fruit. None of the interventions, be they antioxidant vitamins or potent lipophilic antioxidants such as 6-cinnobutol, have succeeded in reducing events in the clinic. So I'm going to stick with my comfort zone, which is cytokines, for much of the rest of this talk. So my history with cytokines goes back a long time, before interleukin-1 beta had that name, and as we'll discuss later, before interleukin-6 had that name. So back in the 1980s in my laboratory, we discovered that vascular cells can make pro-inflammatory cytokines. This was very contrarian and got me in a lot of trouble with the mainline immunology community, because even the name interleukin suggests that these mediators are meant to communicate between properly pedigreed inflammatory cells, leukocytes. But we were able to show that when we stimulated human endothelial cells or smooth muscle cells with a pro-inflammatory stimulus, bacterial endotoxin, lipopolysaccharide, that we were able to induce the messenger RNA that encodes IL-1 beta. We also showed in this paper IL-1 alpha message was increased, and the biological activity in the protein of these pro-inflammatory cytokines were induced. Now, back in the 1980s, we thought that lipopolysaccharide was a laboratory artifice. But now, with our increased appreciation of the importance of the microbiome and impaired intestinal epithelial barrier function, endotoxin may be much more biologically and pathophysiologically relevant than we conceived when we did these experiments. Well, very soon after that, we discovered that IL-1 can induce its own gene expression, shown here in vascular cells. But with Charles Di Norello, who's really the hero of interleukin-1 beta, we showed in vivo in rabbits and in human mononuclear phagocytes that IL-1 can induce its own gene expression, providing an amplification loop. We then discovered that interleukin-1 can markedly increase the expression of another cytokine known as interleukin-6. We were already interested in interleukin-6 for a focused reason that I'll remind you about in a moment, back in the 1980s. This experiment was done by Harald Lappnau, who was my first German postdoctoral fellow and who's now retired. And so I'm facing now how my trainees are retired, but they haven't been able to stop me yet. Okay? We then localized the enzyme that cleaves the inactive precursor of interleukin-1 beta to its mature form, which exerts its biological activity. This is a foam cell actually trying to be a giant cell in human atheroma that is stained for caspase-1, which is the protease that interleukin-1 beta converting enzymes, the other name, that can cleave pro-IL-1 beta to its active form. And we showed back in 1997 that when we stimulated human vascular cells with a stimulus that we knew was operating in human atherosclerosis at the time, CD40 ligand or CD154, we were able to coax the processing of the inactive 33 kD precursor to the 17 kD active form, and that was in a way that was inhibited by interleukin-1 beta converting enzyme inhibitor. So now we've got a biological pathway where we have the inflammasome, which when we were doing our experiments in the biochemistry laboratory, we didn't know that simultaneously that the late Jörg Schopp in his laboratory in Lausanne found that caspase-1 was the business end of this supramolecular structure known as the inflammasome, which senses danger, including from biologically plaque-relevant stimuli such as cholesterol crystals, as shown by Akelat and colleagues, turns on caspase-1 and generates active IL-1 beta. Then IL-1 beta and its cousin, IL-18, are matured, and then we have amplification loop number 1. Then we have amplification loop number 2. Remember that log-log scale that showed the striking amplification of IL-6 production by cells that are stimulated by IL-1. Why were we interested in IL-6 back in the 1980s? Because we already knew that this compound, it was called beta-2 interferon when I started working on this, that IL-6 is the proximate stimulus for what we call the acute phase response. The acute phase response, of course, is how the hepatocyte deals with danger. When we're faced with a dangerous situation, be it trauma or be it pathogens, the hepatocyte switches some of its substrate and energy towards making mediators that are important in host defenses. It says we don't need so much albumin right now. What we need is, for example, fibrinogen to staunch bleeding. We need plasminogen activator inhibitor to block endogenous fibrinolysis because there's a very strong Darwinian survival value to staunching bleeding when we're wounded. Okay? Now, fibrinogen and PI1 are very poor biomarkers for reasons we can go into, but a very high fidelity marker of this acute phase response are the pentraxins, C-reactive protein, or in the mouse we like serum amyloid A. Maybe the mouse likes serum amyloid A. By the way, we're inspired by Jean Davignon's interest in art, and I'm so honored, so special to be here with Rita and to memorialize Jean, who is, of course, a great leader and a great friend. So this is a stained glass depiction of the inflammasome in the long axis view, overlying an artery that is developing an atheroma with foam cells and cholesterol crystals that was executed by Joel Coet and co-designed with him. With Hafid Etoufela, who's now a mid-career person active in this area, we wrote up this piece about targeting cytokines in therapy, and we're able to grace the cover of ATVB with the IL-1 beta ribbon structure, again, a collaboration between me and Joel Coet. So, where did this lead? All of the 20 years of laboratory work led to being able to convince Novartis to sponsor a daring clinical trial. Paul Ricker and I went to Basel in February. I pitched this to Novartis in 2009 in October. In 2010 in February, just about this time of year, we went to Basel and we interacted with the leadership of Novartis, and were able to convince them to embark on this journey with us to actually do an outcome trial with an anti-inflammatory agent targeting IL-1 beta. We enrolled people who had survived a myocardial infarction and who were on what was then the standard of care, but who had lingering inflammation as depicted by above median CRP level. We randomized them to one of 3 concentrations of the monoclonal antibody, canakinumab, why 3 doses in the phase 3 trial. Maybe after 2 glasses of wine tonight, we can discuss that. And at this point, inflammation in atherosclerosis was very controversial. There were many skeptics, and so we powered this trial for hard cardiovascular endpoints, hard MACE, not the expanded MACE, which people often use. You see them there in yellow. And as everyone knows, this study was positive. Now, it's interesting because we've seen and we will see the results of Fourier and Odyssey several times, and I'm always confronted with people saying that the effect is modest. How many papers have I reviewed that calls the benefit in CANTOS modest? But it's precisely the same as the event reduction in the alirocumab and evolocumab studies. So if this is minor, then the whole edifice of PCSK9 is minor as well. In a pre-specified analysis, Paul calculated what happens to cardiovascular mortality and all-cause mortality, the holy grail of all clinical trials, in those who actually achieved a good response to the canakinumab by reducing C-reactive protein greater than to below 2 milligrams per liter. And as you see, there was an over 30% reduction both in CV and total mortality in those who responded well to the drug. Now, we paid a price in terms of impairing host defenses, which is why I mentioned the pro-resolving mediators. In a study of over 10,000 people for 3.7 years of median exposure, we did have a signal for increased infection. Now, and some of these infections were fatal, and it was statistically significant with 10,000 people. But we know who these people are, and we know what the infections are, and I think that some of this risk can be remediated by applying what we learned from this trial. But in contrast, there was striking benefits for inflammatory diseases and other outcomes which have been published. I won't go into them, but just look at gout. Gout was decreased by more than 50%, and gout is, of course, an inflammasome disease with monosodium urate crystals like cholesterol crystals in the plaque. Also, maybe the reason that the Data Safety Monitoring Committee kept the study going despite signal for infection was because of this result, which was adjudicated by a panel of oncology endpoint experts who were impaneled at the outset of the study because there was enough preclinical data to suggest that there might be an anti-cancer effect. That, as you see, at the highest dose of canakinumab in this trial, there was a 77% reduction in fatal lung cancer. So one of the reasons that we don't have canakinumab in our armamentarium today is because the sponsor is off and running, trying to evaluate canakinumab as an adjuvant therapy in lung cancer. Well, we had a win, and it's lucky that our first trial was a win because otherwise the word from, well, let me pick a city at random, Dallas, would have been, you see, it's all smoke and mirrors, the hot air, the inflammation hypothesis. But Dr. Ridker conceived and got funding for another daring trial, which was to apply in cardiovascular patients a modality that has transformed the treatment of rheumatoid arthritis and certain other chronic inflammatory diseases, the cardiovascular inflammation reduction trial, CERT trial, that used low-dose weekly methotrexate. You'll notice that Aruna, who is here in the third row, was very important in the conduct of this study, which proves you don't need big pharma to do trials because this was done on an NIH shoestring, of course, maybe with a little cost sharing, we'll see. And you all know that despite being transformative in rheumatology, that our patients, at least those who were enrolled in this trial, did not respond to methotrexate. And if we compare what went on in CANTOS to what went on in CERT, you see that we had the biomarkers of inflammation going the right direction in CANTOS, but not in CERT. Again, maybe after the third glass of wine, we can go into why CRP was not used as an entry criterion in the CERT trial. So where do we go from here? Well, there's colchicine, and I think you all are aware that there are two large-scale trials, well conducted, the COLCOT trial and those in the early phase post-myocardial infarction, and in the LODOCO-2 study in patients who are in the more chronic phase of atherosclerosis, what we used to call stable, but we don't do that anymore. And you're all aware of those results, and actually, Aruna, you provided the discussion in the AHA for the COLCOT study, and did a wonderful, masterful job of putting this study into context. One of the points is that Remember that these trials were smaller than CANTOS, yet there still is a signal for infection, as you see here under pneumonia, statistically significant increase in pneumonia. So stay tuned for when we have longer exposures and larger number of people, whether or not there's going to be a price to pay for colchicine. Aruna, this probably is reminiscent of what you presented much better at the HA when you did the discussion. Again, there's a small study, the COPS trial in acute coronary events, that actually showed a disquieting signal for mortality, if I remember that correctly, right? No, Aruna, let the record show that Aruna is nodding. Okay. Again, a wonderful discussion for us to have over dinner. So inflammasome inhibitors, there is nary a large pharma or biotech company that doesn't seem to have an inflammasome inhibition program now. Many of us are involved in some of those as well, but it's not ready for prime time. But let's talk about my old friend IL-6. Remember that IL-6 is part of this pathway, and it's actually, based on human genetic studies, the causal part of this pathway in terms of human atherothrombosis. So let's focus on IL-6 a bit. Again, going back to the 1980s, here is a northern blot showing inducibility of IL-6 by IL-1. And I already showed you that log-log plot on the far left here, your far right, sorry, where you see this enormous induction of IL-6 by IL-1. Okay? So where is this going? Well, let's drill down into a subset of patients in whom we don't want to give colchicine, those with chronic renal disease. As I already indicated to you, in my practice and in the practice of many of you, the median eGFR is probably around 45. So we see in our cardiovascular patients, Jorge, you see the same patients I do, most of them have chronic kidney disease. So a very important part of our practice, but we don't want to give colchicine to those people. So we went into the CANTOS database with our nephrology colleagues, Catherine Tuttle and Vlado Perkovic here, and asked the question, what drives recurrent events in those who are well treated with statins? Is it inflammation, residual inflammatory risk, or is it residual lipid risk? And by comparing extreme quartiles in CANTOS, if you look at the 8,000 odd patients in CANTOS who did not have chronic kidney disease, you see that both CRP and LDL correlated well with recurrent events. But if you look in those with chronic kidney disease, the over 1,000, almost 1,100 patients with chronic kidney disease in CANTOS, we saw that although inflammation was still associated with recurrent events, LDL risk was not. So this is a really concordant if we think about the statin literature. Where have statins failed? You know, in Aurora and in 4D, in the statin trials in patients with kidney disease. So these results from CANTOS are really concordant. And so where is this leading? Well, there is a monoclonal antibody which specifically neutralizes IL-6 ligand, ziltivekimab, and with a group of people led by Paul and by Mike Davidson, we did a phase 2 study which was completed successfully during overlapping the pandemic, which I think, Aruna, you'll remind us that Prominent was conducted during the pandemic, and you managed to pull it off by dint of your grit and leadership, and the support of COA, of course. And this was a dose-ranging study with ziltivekimab in individuals with chronic kidney disease who are our target population for reasons I pointed out. And what we see is a striking decrease in the biomarker C-reactive protein. Now we got maybe 40% decrease in CANTOS, but now we're going right to the trigger for the acute phase response, and you see an over 90% reduction in CRP with ziltivekimab in this population. It was sustained. And that is the prodrome to the ZEUS study, which is an outcome study with ziltivekimab in a patient population with chronic kidney disease. Why? Well, large unmet need, high cardiovascular risk, people on dialysis die of cardiovascular disease, a particular biologic state because LDL seems less relevant and inflammation more relevant, and colchicine is relatively contraindicated in our CKD population. So where are we? We are where Sir Winston Churchill made this statement at a turning point in this great Second War. Now, this is not the end. It is not even the beginning of the end, but it is perhaps the end of the beginning. And I think that's where we are in this field. Winston Churchill loved this neighborhood. He wrote passionately about this being a very special place. He was an amateur watercolor artist, and he drew this, you see WSC in the corner there, in the Mamunia Garden, a few steps from where we are sitting today. And here is a little painting of Marrakesh in Churchill's words that he painted looking at the Atlas Range and back, and beautiful Marrakesh in the foreground. I'm so grateful to be part of this. It's an honor to share the podium with such wonderful leaders. And we're here to finish the work, the unfinished work in residual risk. This is where my laboratory works now. And I hope to come back at a future meeting, maybe in Marrakesh, and talk about some of the non-standard risk factors that we think are the horsemen that are driving inflammation, including clonal hematopoiesis, which much of my lab is working on, and I collaborate very well with Alan Tall, who will be telling you about clonal hematopoiesis as an inflammatory stimulus. And these are some of the people in my laboratory who contributed to the work that we're doing, sacrificing some mice in times of COVID. Thank you so much.